Good morning, students. Okay, I'm going to continue with the photosynthesis. Today, I'm going to talk about cyclic and non-cyclic phosphorylation, as well as the dark reactions of the dark reactions of the photosynthesis. Okay, now this one is a plant cell. Okay, where light reactions occurs in thylakoids. Light reaction occurs in thylakoids, where light is used as energy, which photolizes the water molecule, and releases oxygen. At the end of Kelvin cycle, sugar is produced from the carbon dioxide. Okay, and sugar may be further converted into to starch or sucrose. Okay, according to the needs of the plant. Now moving with the photosynthesis. Okay, as I told you, there are two reactions which make up photosynthesis. One of them is the light reaction or a light dependent reaction, and which produces energy from solar power in the form of ATP and NADPH. ATP and NADPH. Okay, and where NADPH further enters ETC, electron transport chain, to produce ATP, to produce ATPs. Now the second part is a Kelvin cycle. It's a Kelvin cycle or light independent reaction, also called as carbon fixation or C3 fixation. It uses energy, ATP and NADPH from light reaction to make up glucose, or sugar, Now the electron flow in the right direction of first one is the cyclic electron, the second one is the non-cyclic one, and the second one is the non-cyclic electron flow. Okay, and this reaction occurs in the thylakoid membrane. Okay, and where the electrons pass from cyclic way and non-cyclic way. Okay, whereas in the case of bacteria, there is only one electron flow. Okay, there is only one reaction cycle. Whereas in the case of plants, cyanobacteria, there are more than one electron cycles. So this is the trapping of sunlight energy, photolysis, by photosynthetic pigments to release chemical energy. The light's energy splits water into hydrogen and oxygen, and the oxygen can be released as a byproduct. The hydrogen then combines with a hydrogen acceptor NAD to produce NADH2. At the same time, energy is also made available to convert ADP plus Pi into high energy molecule ATP, and this process is known as photophosphorylation. Okay, this process is known as photophosphorylation. The NADH2 ATP made by the photolysis are passed to the carbon fixation stage, where carbon dioxide is used as a carbon source, and the photolysis occurs on the grana of the chloroplast. So how photolysis occurs? In the presence of sunlight, water Okay, water molecule is converted into oxygen. Water is converted into oxygen. Okay, NADPH H2 and ATP is produced, which will be used in the fixation of carbon dioxide, which will be used in the fixation of carbon dioxide using Kelvin cycle at the end of the cycle, okay, or in the process of the cycle, a sugar molecule will be produced. Okay, a sugar molecule will be produced. And the photosystems, starting with the photosystems, the photosynthetic pigments such as chlorophyll A or B and carotenoids are light harvesting molecules found in the thylakoid membranes of the chloroplast. The pigments are organized along with the proteins into complexes called photosystems. Each photosystem has light harvesting complexes that contain about 300 to 400 chlorophylls and other pigments. And when a pigment absorbs a proton, it is raised to an excited state, meaning that each one of its electrons is boosted to a higher energy orbital, okay, which helps to transfer electrons to the other carriers of the system. Okay, so most of the pigments in the photosystem act as an energy funnel, passing energy inward to a main reaction center. When one of these pigment is excited by light, it transfers energy to a neighboring pigment through a direct electromagnetic interaction, which is called resonance energy transfer, and this energy transfer is one of the main transfer of electrons which occurs basically in the photosynthesis. Without this, the cyclic electron flow or non-cyclic electron flow will not be possible. Okay, so electrons need to be in excited stage in order to transfer them. So the neighboring pigment in turn can transfer the energy to one of its own neighbors again. Okay, and this process can be repeated multiple of times. In this transfers, the receiving molecule cannot require more energy for excitation than donor, but may require less energy because they are already in the excited form. And collectively, the pigment molecules collect energy and transfer it towards the central part of the photosystem, which is called as reaction center. Now the photosystem, okay, this one is a photosystem diagram of it, okay, and these are the pigment molecules, okay, a chlorophyll A pair, okay, and whole is a light harvesting complex, and this whole is a reaction center, okay, and this one basically is a primary electron acceptor. Okay, now starting with the process, light basically bombards its photons, which is absorbed by the chlorophylls, and chlorophyll A, a special pair, transfers the electrons to the primary electron acceptor. Okay, same occurs in this side also. Okay, the same process occurs in this side also. Okay, so this is how photosynthesis works. Okay, this is just the basics. Now moving more into the photosystem, the reaction center of a photosystem contains a unique pair of chlorophyll A molecules called as special pairs. And once energy reaches the special pair, it will no longer be passed on to the other pigments through resonance energy transfer. Instead, special pair can actually lose an electron when excited, passing it to another molecule in the complex called primary electron acceptor, as I told you in the earlier slide. With this transfer, electron will begin its journey through an electron transport chain. And there are two photosystems present in plants and cyanobacteria. Okay, one is photosystem 2 and the second one is photosystem 1. PS2 comes first in the path of the electron flow, but it is named second because simply it was discovered after photosystem 1. Okay, there is no such any scientific reason behind it. Okay, now some of the basics of the photosystems. Okay, 1 and 2. The special pair. The chlorophyll A special pairs of two electrons absorb different wavelengths of the light. The special pair absorbs best at 680 nanometer of the light, while photosystem 1 absorbs at 700 nanometer. Okay, absorbs at 700 nanometer. Because of this, special pairs are called P680 and P700 respectively. Okay, P680 and P700 respectively. Okay, so PS2 absorbs best at 680 nanometer and PS1 absorbs best at 700 nanometer. Now the primary acceptor, okay, which accepts the electrons which is passed from the chlorophyll A, okay, or any other chlorophyll. The primary electron acceptor of PS2 is a pyophytin. An organic molecule that resembles chlorophyll. Okay, and while a primary electron acceptor PS1 is a chlorophyll called A0. Okay, is a chlorophyll called A0. Okay, and the source of electrons. Once each electron is lost, each photosystem is replenished by electrons from a different sources. For example, the PS2 reaction center gets electron from water, while PS1 reaction is replenished by electron flow down in electron transport chain from the PS2. Okay, so PS2 will receive electrons from water and PS1 from the PS2 through a flow of electrons. Okay, and the main source is through also the excitation of the chlorophyll molecules. So photosystem 1 and 2, okay, this one is a photosystem 2 and a photosystem 1. Okay, light, with the help of light, chlorophyll A will get excited, and the special pair which is known as 680 P680 pair will transfer its electrons to pyophytin, which is basically a primary acceptor. Okay, which is basically a primary acceptor, as well as electrons, more number of electrons will be also provided from the photolysis of water molecule into oxygen, and this for the electrons are passed on to the photosystem 1, and these electrons will be transferred to a special pair of P700, and these electrons will be further passed to the chlorophyll A0, which is the primary acceptor in the case of photosystem 1. So during the light dependent reactions, an electron that is excited in photosystem 2 is passed down to electron transport chain to PS2, losing energy along the way. And in PS1, the electron is excited again and passed down to the second leg of the electron transport chain to a final electron acceptor. Okay, so now let's test the electrons. Okay, simply this one is the electron transport chain for the photosynthesis. how ATP will be produced. Okay, how ATP will be produced after the photosynthesis. Okay, and chloroplast. Okay, now with the help of light, okay, chlorophyll gets excited. A P680 pair, P680 pair transfers electrons to the first electron acceptor, which is a PO15, a hydrogen will be released. Alongside, electrons will be also passed through the photolysis of water molecule. Okay, and where water will be converted into oxygen, and these two hydrogen ions are released into the interior of the thylakoid lumen, and passing further to the chain, the electrons will be transferred to the P700 photosystem to PS1 P700 complex, where it will be excited again in the presence of light. And these electrons will be further passed to the electron acceptor chlorophyll A0. Okay, and these electrons will be further passed on to the formation of NADP into NADPH, and this NADPH will further move electrons to the electron transport chain, and at the same time a hydrogen gradient will be produced in the interior of the thylakoid lumen. Okay, in the interior of the thylakoid lumen, where with the help of enzyme ATP synthase, converts ADP into ATP. The mechanism is same we studied during chemiosmosis. Okay, this mechanism is same which we studied during chemiosmosis. Okay, so I am repeating the whole process again. P680 gets excited where a pair of chlorophyll A transfers electron to the primary electron acceptor PO15. At the same time, with the help of photolysis, water molecule will be converted into oxygen, and these electrons are further passed on to the PS1, where the reaction center is P700. At the same time, hydrogen will be further added to the thylakoid lumen in the interior part, and the P700, where electron will be further excited with the help of light again, and these electrons will be passed on to the chlorophyll A0 and move forward to produce NADPH from NADP plus, and further electrons are passed on to the electron transport chain. Okay, and through the process of chemiosmosis, ATP synthase will produce ATP from ADP. Okay, from ADP. Now this one is a free energy graph with reaction at various stages. Okay, P680 photosystem 2, light energy, water which also provides electrons and will be passed on to the PO15 through a series of reactions. Okay, you can see here clearly the free energy of the electron decreases. Free energy of the reaction of the electrons decreases. Now again with the help of light energy, electrons will again get excited and passed on to the chlorophyll A0, where finally it will be passed on to the formation of NADPH. This is how a variation of free energy of electrons occurs with the progress of progression of the photosynthetic reaction. Now the cyclic photophosphorylation. Okay. Now the cyclic photophosphorylation. Okay, as I told you earlier also, I will discuss cyclic photophosphorylation and non-cyclic photophosphorylation. Now the cyclic photophosphorylation basically starts when photosystem 2 provides electrons, okay, electrons, and these electrons are further excited and passed on to the primary electron acceptor, okay, and this further again are passed to the other carrier molecules of the photosynthesis So this whole process is a cyclic process. Okay, it's a cyclic process. Thus, this is the reason why it is called as cyclic phosphorylation. And rest of the electrons which are passed on further to produce NADPH, and electron transport chain through which, as well as the chemiosmosis, which produces ATP, we almost said, which we have studied earlier. Okay, now the cyclic electron flow occurs in the thylakoid membrane and uses photosystem 1 only, and P700 reaction center chlorophyll A uses electron transport chain, generates ATP only. Okay, generates ATP only. Now the cyclic electron flow, okay, is somewhat like this one. It's somewhat like this one. Okay, it with the help of accessory pigments, photosystem 1 is passed on to the primary electron acceptor. Okay, these electrons are further passed through various carriers of the system. Okay, and the pigments basically absorb light energy, excite electrons of chlorophyll A to produce ATP. Okay. Now the non-cyclic electron flow, okay, occurs in the thylakoid membrane, uses photosystem 2 and photosystem 1, where P680 reaction center is photosystem 2 and P700 is a reaction center of photosystem 1, uses electron transport chain, produces oxygen, ATP and NADPH. Okay, and NADPH. Now the non-cyclic flow. Okay, so photosystem 2, okay, where electrons get excited and are passed to the primary electron acceptor, and through which further move to the ETC, and ATP will be produced and passed on to the photosystem 1 P700, and the primary electron acceptor, which is a chlorophyll A0, will now be further be passed on to produce NADPH. So there is no cycle. Okay, this is simply a process which moves on further. Okay, Okay, and in the process, I am again repeating, the H2 is split in the PH2 and ATP is made, while the energy carrier NADPH is made in photosystem 1. So in the non-cyclic ones, ADP will be converted into ATP, NADP plus H will be converted into NADPH, and oxygen comes from the splitting of of the water, not the carbon dioxide. Okay, so what is the function of carbon dioxide? That the carbon dioxide will be converted into a sugar molecule, okay, in the dark reaction, the Calvin cycle, but not during the photo reactions. Okay, now chemiosmosis, I'm just speeding up this process as we already studied. Okay, it's a process which powers ATP synthesis, takes place across the thylakoid membrane, uses electron transport chain and ATP synthase, which is basically, as you know, it's an enzyme. and hydrogen move down their concentration gradient through a channels of ATP synthase, forming ATP from ADP. Okay, forming ATP from ADP. Okay, so this is the whole process of the chemiosmosis. The whole process of the chemiosmosis. Okay. Now, Kelvin cycle. Okay. Now, the Kelvin cycle. The carbon fixation, basically, which is a light independent reaction, and C3 plants, which are almost 80% of the plants of the earth, and it occurs in stroma. Okay, it occurs in stroma and uses ATP and NADPH from light reaction as energy, from light reaction as energy, and uses carbon dioxide for carbon and to produce glucose it takes about 6 tons and uses 18 ATPs and 12 NADPH and 12 NADPH. Okay, as it turns out, the atoms of carbon in your body were once the part of the carbon dioxide molecules in the air. The carbon atoms end up in you and in other life forms, and thanks to second stage of photosynthesis, which is basically known as Kelvin cycle, also known as carbon fixation stage, also known as the dark reaction. And this stage is a series of enzyme controlled chemical reactions in the form of a cycle, Okay, as the carbon dioxide enters carbon fixation, it combines with the 5-carbon ribulose biphosphate to form an unstable 6-carbon compound, which immediately splits up into 3-carbon glyceride phosphate. Okay, so starting with the process of the Calvin cycle, as the carbon dioxide enters carbon fixation, it combines with a 5-carbon ribulose biphosphate to form an unstable 6-carbon compound, which immediately splits up into 2 molecules of carbon glycerate phosphate. Okay, of a 3-carbon glycerate phosphate. Okay, this is a place where Kelvin cycle occurs. Kelvin cycle occurs in the stroma. It occurs in the stroma. Okay. Now Kelvin cycle. Through the light reactions, NADP, H, and ATP are provided to the Kelvin cycle. Where carbon dioxide is basically converted into phosphoglycerate. Okay, and from glycerate 3-phosphate it will be converted into sugars. Okay, so moving further with the Kelvin cycle in a more detailed form, with the help of enzyme Rubisco. Okay, with the help of enzyme Rubisco, an unstable 6-carbon compound is formed. Okay, which will be converted into phosphoglycerate. Okay, and this will be further used to produce glucose, and rest of the glycerate phosphates are further converted into ribulose biphosphate ribose biphosphate again with the help of enzyme will be again converted into a 6 carbon unstable compound. So in the process, as I told you earlier, okay, many number of ATP molecules will be utilized during this process. Okay, many number of ATP molecules will be utilized in the process. Okay, now this one is a whole. Okay, this one is a Calvin cycle where C3 fixation occurs. Okay, well, the C3 fixation occurs. So as I told you, about 6 cycles occur in the Calvin cycle. In the Calvin cycle, and remember, okay, C3 for the Calvin cycle where glucose will be produced. Okay. Now photorespiration. Okay, and how does the photorespiration occurs? It occurs on hot, dry, and bright days, and stomata is closed, and fixation of O2 instead of carbon dioxide, and produces 2 carbon molecules instead of 3 carbon sugar molecules, produces no sugar molecules or no ATP Okay, because of the photorespiration, plants have special adaptations to limit the effect of photorespiration because it can be harmful to some of the plants such as C4 plants as well as CAM plants. Now the C4 plants, what are the characteristics of the C4 plants? They grow in hot, moist environments. Examples are grasses, corn, sugarcane, etc. Okay. Photosynthesis occurs in 2 places. Okay, light reaction mesophils, and in Calvin cycle occurs in bundled sheath cells. Okay, bundled sheath cells. Okay, this is a photograph, as you all know what is this. Now C4 plants, which occurs in the photosynthesis occur in the mesophyll cell. Okay, now this phosphopyruvate, not that one. The malate will be produced. Okay, and which will be further passed on to the carbon dioxide, which then will be converted into glucose, and glucose will be passed on to the vascular tissue. Okay, and also in the process pyruvic acid will be also produced. Okay, now CAM plants, what are the characteristics of the CAM plants? They grow in hot, dry environments. 5% of the plant cactus and ice plants. Stomata are closed during the day in order to save water. Okay, and stomata opens during the night, and light reactions occurs during the day, and Calvin cycle occurs when the carbon dioxide is present. Okay, so in the CAM plants where in the night stomata opens. Okay, when the CAM plant stomata opens, in the presence of carbon dioxide, it will be converted into malate. Okay, and passed on to the vacuole. Okay, and the day when stomata are closed, why stomata are closed? In order to reduce loss of water. Okay, malate will be converted into pyruvic acid, and the carbon dioxide which is released will be further passed on to produce glucose. Okay, so in the whole process Okay, when stomata are open, when stomata are closed. Okay, pyruvate, which we convert in starch into triphosphate, into phosphoryl pyruvate. Okay, by the addition of HCO3 from where carbon dioxide enters, gets converted into oxaloacetate with the help of enzyme PEP carboxylase, where oxaloacetate will be converted into malate. Okay, with the help of enzyme NAD malic dehydrogenase will further passed on to the vacuum where malic acid will be produced. And further will be converted into malate, where malate with the help of NADP malic enzyme produces pyruvic acid and carbon dioxide will be released. Okay, and this carbon will be taken up by the Calvin cycle to produce carbohydrates. Okay, so why do chem plants close their stomata during the day? Okay, as I told you earlier also, chem plants close down their stomata in the hottest part of the day to conserve water. Okay, so C3 plants use C3 pathway. Okay, this is a diagrammatic representation. If you want, whichever diagram you want to use, you are free for that. Okay, and the C4 ones use C4 pathway. Okay, C4 plants use C4 pathway. This is a diagrammatical representation. Whichever diagram you want to use, you are free to use. Okay, whichever you find easy, you can use that diagram for your exam. Okay, now which are the limiting factors of the photosynthesis? Okay, which are the limiting factors of the photosynthesis? Chemical reactions such as photosynthesis can be speeded up or slowed down depending on certain conditions. A conditions which is able to limit rate of a reaction is usually a limiting factor. Okay, and these limiting factors can be studied with the help of a Eldia bubbler. Where Eldia is a special plant which grows in the water. Okay, through which we can study the effect of light, carbon dioxide concentration, temperature, and one at a time to show that they are all limiting factors. Rate of photosynthesis can be measured by counting the bubbles of oxygen released in a specified time. Okay, you can also perform this experiment at your home also if you want. Okay, so this is a diagram. Okay, this is an diagram if you want to perform at home. Okay, which is a very easy experiment. Okay, here lamp will be used. Okay, lamp will be heat shield. Because why heat shields will be used? If you do not use heat shield, then due to the light, this may heat up. Okay, this may heat up. So here glass should be used as a heat shield so the heat do not pass through this beaker. And a dilute sodium bicarbonate is added in order to produce carbon dioxide. And in this funnel we have LDO plant. Okay, and after some time you can clearly see the oxygen bubbles which are produced. Okay, at the end of the experiment. Okay, this is not a short process which occurs in 15 minutes. It may take up to hours. Okay, depending on the conditions, and we can vary amount of carbon dioxide, light, etc. Even temperatures also. Okay, so this experiment. So temperature, light intensity, carbon dioxide concentrations are the possible limiting factors of the rate of the photosynthesis. At a high level of light intensity, further increases and have no effect on the rate of photosynthesis. Therefore, light intensity is not the limiting factor, but another factor, that is temperature, carbon dioxide concentration, enzymes or chloroplast working at maximum efficiency is a limiting factor for photosynthesis. Okay, but there are some conditions when a very high amount of light can also have harmful effect on the chloroplast. Okay, because of that the temperature increases, which leads to breakdown of various segments of the plants. Okay, which may further also reduce the photosynthesis. But by increasing light, simply you cannot increase rate of a photosynthesis, but up to some extent you can increase, but not you simply go on and increasing light intensity does not mean any. At a time you will see simply a straight line, okay, a plateau like structure. However, how much you increase light, there will be no difference in the rate of the photosynthesis. Now carbon dioxide concentration, okay, and rate of photosynthesis. Carbon dioxide is a substrate for the metabolic pathway. Hence, the relation should be similar to how enzyme reactions are limited by a substrate concentration. As you know, in order to move forward with enzymatic reaction, substrate is a limiting factor. So in this case also, where carbon dioxide is a substrate for the Calvin cycle and its enzymes, in the absence of carbon dioxide, the Calvin cycle will not be able to move forward. Okay, when the carbon dioxide concentration is increased, the rate of photosynthesis increases similar to light, but also it is a limiting factor at low concentration similar to light. But up to some extent, the whole process cannot increase. It's simply a straight line. Okay, so this plateau will be formed. Okay. Now the temperature. Okay, temperature depends on the type of plant and type of the geography where plants are grown and their native environment also matters. Okay, you can simply cannot grow a plant of cold regions into tropic regions. Okay, so some of the plants which are in let's say are near the Arctic so in the such as Russia, colder regions of Canada, etc. Those plants cannot be grown in India, especially in Gujarat. Okay, so temperature is an important limiting factor, and the photosynthesis may vary according to the optimal temperature needs. Okay, the photosynthesis is a metabolic pathway. Okay, and which is dependent on enzyme reactions which are affected by temperatures as well as carbon dioxides. Okay, so the increase in the temperature give molecules more kinetic energy, causing substrate to collide with the active sites more frequently, and this increases the rate of photosynthesis. And now you can see clearly, okay, at the optimal temperature, rate of photosynthesis increases. But if we go on increasing the temperature more than optimum, the rate of photosynthesis will also decrease. As the temperature approaches the optimum, the enzyme begins to denature to become non-functional, causing the rate of photosynthesis to increase and more slowly eventually to the peak. After the optimum temperature enzymes denature, the rapidly causing fast decrease in the photosynthesis occurs, as I told you. Okay.